Hello, everyone. Welcome to our monthly Science of Alt Protein seminar. My name is Amy Huang, and I'm the Senior Associate Director of Scientific Ecosystems at the Good Food Institute. For those of you who are here for the very first time, welcome to our community. The Good Food Institute is a nonprofit think tank that is 100% driven by philanthropy, and our singular goal is to build a world where alternative proteins are no longer alternative. We see scientists like all of you as really critical to that mission, so thank you for being here to learn about some neat research happening in the field. If you are looking to take a deeper dive into the science of plant-based fermentation or cultivated meat technologies, we've got a whole lot of open access resources that you can find at gfi.org/science. And now I'm very excited to introduce today's speaker. Dr. André Damasio is an associate professor in the Department of Biochemistry and Tissue Biology at the Institute of Biology at the University of Campinas, also known as UNICAMP in Brazil. He received his Ph.D. in biochemistry from the University of Sao Paulo in 2011, with research periods at Oklahoma State University and the Institute of Catalysis and Petroleochemistry in Madrid. He joined UNICAMP in 2015 as an assistant professor after completing his postdoc at the Brazilian Biorenewables National Lab, and his research group is dedicated to studying microbial biomanufacturing, mainly the production of recombinant proteins and enzymes using filamentous fungi as cell factories. Dr. Damasio, we are so excited to have you here with us today, and I know our community is really eager to learn from you. So I will turn the floor over to you.
Hey, all right. Hi, everyone. Thank you, Renee, for inviting me, and Amy for the kind introduction. And thank you also the GFI Brazil for the very nice work you have done. And it's a great opportunity to present our research at this GFI seminar. As Amy said, I'm associate professor at the University of Campinas in Brazil, and I lead the laboratory of metabolic engineering of microorganisms. And today I'll be discussing our approach to glycoengineering fungal cell factories and implications for recombinant protein production. Let me outline what we'll cover today. First, we will begin with the fundamentals of microbial biomanufacturing. Next, I will focus on recombinant proteins and cell factories. And finally, we will focus on what I consider the central topic here, the glycosylation challenge. Then I will present 3 studies from our lab that demonstrate an approach to glycoengineering filamentous fungi. First, we mapped the native glycoproteome of a fungal strain to establish our baseline. Second, we redesign individual glycosylation sites in a specific recombinant protein to understand their functional impact. And finally, we use CRISPR-Cas9 to create pathway-level glycoengineer fungal strains. Let's start with the fundamentals. Microbial biomanufacturing can be understood through 3 interconnected pillars. The upstream phase involves designing both the microbe and the target bioproduct, in our case, recombinant proteins, which includes strain engineering, gene optimization, metabolic pathway design, and host selection. In the fermentation phase, focus on growing the microbe through bioreactor operation, process optimization, and scale-up strategies. And finally, in the downstream phase involves purifying the final product through protein separation, purification steps, and quality assessment. What is critical to understand is that these pillars are not independent. decisions made in the upstream phase can have a significant impact on downstream processing costs, purification complexity, and even product quality. We are interested on producing, as you see, in producing recombinant proteins using filamentous fungi as host cells. Recombinant proteins serve as the, how can I say, the building blocks of life and industry. We can categorize them based on function, for example, structural proteins such as collagen and elastin that provide mechanical support, enzymes including amylases and proteases that catalyze biochemical reactions, and signaling proteins, including hormones and growth factors that coordinate cellular communication. From the industrial point of view, the applications can be found in multiple sectors. In medicine, for example, with therapeutic proteins and vaccines, in industry with biocatalysts and detergents, in food and nutrition with alternative proteins and enzymes, in agriculture with biopesticides, and in materials with bioplastics, just to mention some examples. This diversity explains the exponential growth we are seeing in the recombinant protein market and why production efficiency is so critical. But how exactly are these recombinant proteins produced? Recombinant proteins can be produced using microbial cell factories and other cell factories. Independent of the cell factory, the production process follows four fundamental steps. First, gene cloning to identify and isolate the target gene. Second, vector construction to insert the gene into a plasmid. Then host transformation to introduce the genetic material into the host. And finally, the production and purification to scale up and purify the protein. The advantages are scalability, cost effectiveness, consistency, and safety depends on the host cell and the process, of course. But cell factories are not limited to microorganisms. Mammalian cells, such as CHO or CHO cells, and insect cells have also been used for specific applications. CHO cells have been used for the production of therapeutic proteins, including hormones and monoclonal antibodies. And the choice of the system becomes more critical when considering the complexity of PTMs or post-translational modifications that are required for proper protein function. But let's focus on microbial cell factories. This overview illustrates the diversity of microbial cell factories. Each system has distinct capabilities and limitations. Bacteria like E. coli are fast and straightforward, but they lack complex post-translational modification machinery. Yeasts such as Saccharomyces cerevisiae or Deformer Pichia pastoris offer more sophisticated processing, but still have limitations in their PTMs. Filamentous fungi are powerhouses for protein secretion, but they produce many proteases that can degrade your recombinant protein, and PTMs differ from those in mammalian systems, for example. The key insight here is that the choice of host organism should be driven by the specific requirements of your target protein and process, such as titer or its post-translational modification needs. Then different organisms excel at producing different types of proteins, and understanding these capabilities is essential for successful protein production. So why PTMs must be considered? Production of recombinant proteins alone is not enough. Proteins must be correctly modified to function properly. And PTMs are essential for at least four key reasons. First, protein function. They are required for enzymatic activity and binding specificity. Stability. They protect against degradation and misfolding. Localization. They direct proteins to correct cellular compartments. And biological activity, they enable proper interaction with other molecule and biological systems. PTMs are not only decorations, they are functional. That's the point. And this becomes particularly important. And we are producing mammalian proteins in microbial system that have evolved different PTM machinery. Let me focus on one particular PTM, the glycosylation. The N-glycosylation involves the covalent attachment of carbohydrate chains to asparagine residues in proteins, occurring at the consensus sequence you can see here. Asparagine X can be any amino acid, but not proline, followed by serine or threonine. The process occurs at the in the endoplasmic reticulum, the ER, involves the oligosaccharide transferase, an enzyme that transfers the glycan, this core glycan, in block to the nascent protein. This is the core N-glycan present in fungi, composed by 2 N-acetylglucosamine or glycans, 9 mannoses, and 3 glucoses. This is the core sequence in fungi and in mammalian cells found in the ER. I mentioned that PTMs are not only decoration. These figures illustrate the multiple roles of glycosylation. Glycosylation is not only structural, it's fundamentally functional. It affects how proteins fold, their stability, where they are located in the cell, how they are recognized by immune system, and how they perform their biological functions. Now, here's where the challenge becomes clear, I would say. This comparative analysis reveals that the final glycan structures exhibit significant variation across species. High-mannose glycans are commonly found in yeasts and filamentous fungi, while mammal cells synthesize complex structures, as you can see here. Again, this becomes particularly important when we are producing mammalian proteins in microbial systems. And this variation can have real consequences for recombinant protein function, stability, and biological activity. This brings us to what I call the glycan divide, the fundamental difference between fungal and mammalian glycosylation. Fungi, as I mentioned, produce high-mannose type glycans, undergo minimal processing in the Golgi, and can be recognized as foreign by mammalian immune system depending on the context. In contrast, mammals produce complex type glycans with branched, complex structures that contain N-acetylglucosamine or glycan, galactose, and sometimes sialic acid. Undergo extensive processing in the Golgi and is tissue specific in mammalian systems. This divide can have real implications for recombinant protein function and applications. These pathway diagrams show how fungi and mammals share a common basic structure in the ER, but diverge in the Golgi. Fungi add more mannose residues through the so-called hypermannosylation, while mammals trim the mannose and add complex branching structures, as I mentioned before. One solution to the glycan divide has been the so-called humanization of fungal strains. In other words, glycoengineering of fungal cells to produce more mammalian-like glycan structures. A good starting point would be the synthesis of N-glycans containing 5 or 3 mannoses, as you can see here, instead of hyper-mannosylation. And humanization is not just theoretical, it's technically achievable. In this work, published some years ago by Professor Ulf Mortensen's group at the Technical University of Denmark, the N-glycan composition of proteins produced by these filamentous fungi, Aspergillus nidulans, was mapped. They found that the composition of N-glycans ranges from 5 to 10 mannoses, confirming the high-mannose pattern typically observed in fungi. Then they constructed a single and a triple mutant by deleting the ALG genes involved in the N-glycan synthesis. The result was an N-glycan pool with truncated structures, although many of these glycans still contain more than 5 mannoses. Then additional strain optimization was required, in addition to gene deletions. Combining gene deletion and overexpression of mannosidases in the Golgi, they achieved approximately 80% of glycans with 3 to 5 mannose residues in Aspergillus nidulans, closer or much closer to mammalian patterns than the native fungal hyper-mannosylation. However, this work after this glycoengineering did not explore the effect of glycan truncation on fungal cell biology and on recombinant protein function and productivity. Then, if the glycoengineering of fungal cells is possible, I want to discuss a dual impact of glycosylation for recombinant protein production. Our interest is in this side, the structure, function, and productivity. There are many studies showing that glycosylation affects protein function. Our question was if glycoengineering affects recombinant protein production and function, and how these modifications can impact the fungal cell. On the other side, regarding immunogenicity and safety, it is true that high-mannose glycans can be recognized as PAMPs or pathogen-associated molecular patterns and may trigger the immune response. However, this issue is primarily relevant for therapeutic proteins produced by microorganisms because therapeutic proteins are generally injected in the body where direct exposure to the immune system can amplify such effects. In contrast, for food applications, this risk is largely theoretical as ingested proteins are subject to extensive proteolytic and metabolic processing throughout the gastrointestinal tract. Then, I've included here, but it's not the driver of our research. Although I have no data on alternative proteins to share, I would like to illustrate this glycosylation concept by showing you 3 alternative proteins produced in microbial cell factories, which represent different levels of glycosylation. First, beta-lactoglobulin, the easy case. Ovalbumin, this is our moderate case, and kappa-casein, this represents the high complexity case. Let's examine each of these alternative proteins in detail. Beta-lactoglobulin is a 18 kilodaltons protein with a beta barrel structure. No glycosylation is required for proper folding or function, and high expression yields is possible in fungal system. Functional properties are preserved in recombinant form, and multiple commercial products are being developed. This is the easy case. Of albumin, it's a more complex case. This 45 kilodaltons protein comprises 54% of chicken egg white and serves critical functions in foaming, gelling, and emulsification. There is a single N-glycosylation site in this protein at the asparagine 292, and the absence of this glycosylation can affect functional properties like heat stability. And the more complex kappa casein, a higher glycoprotein complexity, it has multiple O glycosylation sites and one potential N-glycosylation site. Around 60% of molecules are glycosylated, and it contains sialylated glycans that are critical for function. Fungal systems lack sialylation capability, as I mentioned the glycan divide slide, and micelle formation and other functions depends on the glycosylation pattern. Okay, we also choose Aspergillus nidulans as our model system for technical reasons. It's a well-established genetic model with a complete genome sequence and extensive genetic tools available. And it demonstrates efficient protein secretion for functional studies at least, and CRISPR-Cas9 tools are available. This is our specific strain, Aspergillus nidulans A773. Our group has achieved success using this strain for the production of many recombinant enzymes, especially carbohydrate active enzymes from different families, as demonstrated in this gel. Given this context, we have developed 3 studies over the last few years. First, we mapped the native glycoproteome of Aspergillus nidulans by characterizing the N-glycosylation sites and glycan structures to establish a baseline for engineering efforts. Second, we engineer individual glycosylation sites to understand their impact on recombinant protein secretion and function. Third, we remodel the glycosylation pathway using CRISPR-Cas9 to delete specific genes, creating fungal strains producing truncated N-glycans. Let me start with this work where we mapped the N-glycoproteome of Aspergillus nidulans. We employ a glycoproteomic and glycomic approach, combining mass spectrometry-based proteomics with glycan analysis. This allowed us to identify which proteins are glycosylation in the secretomes of Aspergillus nidulans, or the pool of secreted proteins, in the presence of glucose and more complex carbon sources such as sugarcane bagasse and xylem. Where the glycosylation sites were located, we can identify, and what types of glycan structures were attached to these proteins. The first key finding here was about the secretome composition. Carbohydrate active enzymes, or cazymes, make up more than 50% of the secreted proteins when Aspergillus nidulans grows on complex substrates. These enzymes have significant commercial value for industrial applications, particularly in the biofuel sector. Therefore, it's relatively straightforward to produce a cocktail of cazymes using Aspergillus strains. Our analysis identified a total of 182 N-glycosylation sites across 103 proteins. Notably, 1-to-1 N-glycosylation sites were found on 67 different cazymes. Then cazymes are heavily glycosylated with 66% of all glycosylation sites identified. We also noted a preference for the glycosylation sequence NXT over NXS, indicating a bias toward threonine over serine in the glycosylation consensus sequence. And the glycan structures were also characterized. We confirmed that the glycan profile is high-mannose, as you can see here, with N-glycans composed mainly of 5 to 9 mannose residues, minimal processing beyond the ER, and consistency across the different carbon sources we test. This confirmed again that Aspergillus nidulans follows the typical fungal glycosylation pathway, producing high-mannose structures that differ from mammalian complex glycans. Okay, and which glycoprotein did we study? This enzyme called beta-xylosidase, or BXLB, was identified among the cazymes in the Aspergillus nidulans secretome as a model glycoprotein for further studies. It's approximately 83 kilodaltons with 7 predicted N-glycosylation sites and enzymatically tractable for functional analysis. This became our model for understanding how individual sites affect protein function. Let me summarize the key conclusions from this first study. Aspergillus nidulans has a diverse substrate-dependent secretome with cazymes dominating on complex substrates. There is extensive N-glycosylation of cazymes, 182 N-glycosylation sites identified, confirming that many secreted enzymes are glycosylated. And secreted proteins carry high-mannose N-glycans, confirming the glycan divide. In this second study, we asked a fundamental question: what happens when we remove individual glycosylation sites from the recombinant protein? Are all glycosylation sites equally important for the protein? We maintain the BXLB as our model glycoprotein, which has 7 N-glycosylation sites. And comparing this Aspergillus nidulans sequence with many fungal homologs available in public database, we observed that the glycosylation sites 1, 5, and 7 were highly conserved. Then we started the mutations with these 3 conserved sites. We designed a series of glycomutants, as you can see here, maintaining the 3 conserved sites. Then we remove individual sites and in different combinations. We also designed a non-glycosylation BXLB, non-glycosylated BXLB, sorry, and even a mutant version with 4 non-native N-glycosylation sites. And this approach allowed us to understand how different sites contribute to overall protein function and secretion. All these mutant versions of PXLB were cloned into this plasmid, the PXpire we have used for many years, under the control of a strong promoter, a cyanopeptide from glucoamylase that is a highly secreted enzyme, and a histidine tag for protein purification. The recombinant strains of Aspergillus nidulans were cultivated in the presence of maltose for BXLB production, and the protein relative concentration were determined by Western blot using a specific antibody for BXLB. We found that N-glycosylation sites have differential effects on protein secretion. As shown here, the native glycosylation pattern was optimal for secretion. All the glycomutant versions of BXLB showed a lower secretion. The mutant which we had non-native glycosylation sites I mentioned was not secreted at all, as you can see here. If you look at some glycomutant secretion over time, it's clear that the native glycosylation pattern was optimal for secretion. This finding demonstrates that glycosylation engineering needs to be strategic. You cannot simply remove all the glycosylation sites. You need to understand which sites are important for folding and secretion of your target recombinant proteins. All the BXLB glycomutants were purified for from recombinant strains of Aspergillus nidulans, and we discovered that removing glycosylation sites could improve catalytic efficiencies, as you can see here. For example, this mutant maintaining the N-glycosylation sites 1, 5, and 7 exhibit 1.5-fold higher catalytic efficiency than the wild-type enzyme. This demonstrated a trade-off. While the native glycosylation pattern was optimal for secretion, it was not necessarily ideal for catalytic activity. This second study showed that the effects of N-glycosylation sites on protein secretion and function depend on their specific positions. While the native glycosylation pattern optimized secretion, it didn't maximize catalytic activity, indicating potential for improvement through rational engineering. Moreover, removing certain glycan sites can enhance enzyme performance in the case of BXLB. Finally, our third study took a different approach. Instead of modifying individual sites, we asked what happened if we modify the glycosylation pathway as previously reported. During my introduction and context, I presented a published paper demonstrating the effective truncation of N-glycans chain by gene deletion, achieving 80% of mannose 3 to 5. Here, the primary question was, what would happen to BXLB, our model glycoprotein, secretion in a fungal cell producing truncated N-glycans instead of native ones? We target the same genes in the N-glycan processing pathway using CRISPR-Cas9 technology, allowing us to construct a double and a triple mutant, removing the enzymes responsible for adding mannose residues into the ER. Here you can see the mutant strains in a standard condition, minimal medium, 37 degrees, 72 hours, which exhibit reduced radial growth compared to the reference strain, demonstrating that glycoengineering can affect cell physiology. Then we transformed the same model recombinant enzyme, BXLB, with the 7 glycosylation sites into these 3 mutants to evaluate activity and secretion. The secretome of the double deletion mutant showed a significant increase in BXLB activity. Evidenced after normalization by total protein secreted and fungal biomass. While the activity in the triple mutant secretome was similar to the reference strain. To determine if this increase in BXLB activity was due to higher secretion, we measured the relative titer by Western blot. And you can see here a 25 to 30% increase in BXLB secretion in the double mutants. It showed that the double deletion positively affected BXLB secretion, resulting in higher activity in the secretome. Then we purify the recombinant BXLB from these three strains for biochemical analysis. The optimal conditions for BXLB produced by all strains were 60 degrees and pH 5. And analyzing the kinetics show that BXLB produced by the double mutant maintained the catalytic efficiency, while in the triple mutant, a 24% reduction was observed. So for the double mutant, it was possible to achieve better secretion without compromising enzymatic function. This provided a proof of concept that pathway level engineering could improve some recombinant proteins production. The next question was if the secretion of native proteins were also affected by glycoengineering. We also observed broader effects on the secretome. The secretomes of the reference strain and mutant strain, the double and the triple mutants, were produced in the presence of pretreated sugarcane bagasse and analyzed by mass spectrometry. Sugarcane bagasse and other types of plant biomass induce the secretion of many enzymes, particularly cazymes used in Aspergillus. Then, if you look here, the secretome data, the abundance of secreted proteins was similar among the strains, but the secretome profile was affected. 34 and 45 unique proteins were found in the double and triple mutant secretomes. And at the individual level, a big cluster, you can see here the reference strain, the triple and the double mutant, a big cluster of down-regulated proteins was observed in the double mutant secretome. We observed few proteins that are more abundant in this mutant than in the reference strain, suggesting a cleaner background of unwanted proteins that could facilitate downstream processes. Then from study 3, we reinforced the application of CRISPR-Cas9 for precise glycoengineering in Aspergillus nidulans. We achieved recombinant protein secretion through pathway level modifications and a potential simplification of downstream processing because the cleaner background. To summarize, it is possible to use fungi glycoengineering to produce at least more mammalian-like proteins. The studies are more advanced for yeasts than filamentous fungi, but we have advanced on that. If you are using a non-well-characterized cell factory, mapping the glycoproteome to identify native glycosylation patterns is fundamental. Second, it is possible to modify specific glycosylation sites and optimize protein function, but the secretion can be affected. This is important. Finally, truncated glycans can enhance the secretion of your target protein, thereby maintaining its function. Before I finish, I would like to share this groundbreaking work published 2 or 3 months ago by Nathan Lewis group from UCSD. It's a nice demonstration that N-glycosylation is a critical determinant for recombinant protein secretion. In this study, they use CHO cells, but not fungal cells, but it's still very interesting work. To share the data in one unique slide, they analyze more than 2,000 human secretome proteins expressed in CHO cells. And as I mentioned, CHO cells have been largely used for therapeutic protein production, such as monoclonal antibodies. And they found that proteins features account for around 15% of secretion variability. And N-glycosylation is listed in the top 3 features together with molecular weights and cysteine content. This large-scale analysis, yeah, as I mentioned, more than 2,000 proteins tested, provides evidence that N-glycosylation must be considered when engineering cell factors for recombinant protein production. And PTM's post-translational modification capacity is a key determinant of secretion efficiency. Before I conclude, I want to point out some limitations of our work. First, we used Aspergillus nidulans as a model system. While this was chosen for its well-characterized biology, it's fundamentally important to extend these studies to industrial strains such as Aspergillus niger and Aspergillus oryzae, which are used in commercial protein production. What works in our model system may not translate to industrial applications. Second, we focus primarily on beta-xylosidase as a model glycoprotein. While this provides insights, we cannot assume the same principles apply to other recombinant proteins. Different proteins may have different glycosylation requirements, different sensitivities to glycan modifications, and different structure-function relationships. Each target protein may require its own optimization strategy. Third, pathway engineering achieved truncated glycans, but we haven't yet achieved full mammalian-like complex glycans with branching, galactose, and so on. The gap between truncated high-mannose and fully complex glycans remains a technical challenge in filamentous fungi. And fourth, all our studies were conducted at laboratory scale. We don't yet understand how this glycoengineering modification affect large-scale fermentation performance or productivity under industrial conditions. Thank you so much. This is a collaborative effort involving multiple institutions and researchers across Brazil, and my colleague Uffe Mortensen from Denmark. I want to acknowledge the extensive collaboration with all partners listed here, the former and current members of the lab, especially Marcelo, Jacqueline, and Ariadne, who obtained all the data I've shared today, and the support from our funding agencies. Thank you very much for your attention. You can find me in the LinkedIn or here in this QR code personal website. And thank you very much for your attention. I'm happy to take your questions.
Thank you so much, Dr. Damasio. That was amazing. This is not my field of study, but still super fascinating, and I learned a whole lot. Definitely comes through that there's a lot of promise here to improve functional ingredients for our field and then also to lower production costs in a really major way. So Thank you for sharing your work with our community. Feel free to stop sharing your screen at this point. And for those of you in the audience, if you haven't yet gotten the chance to, please drop your questions into the Q&A section of your Zoom bar, and we'll start working through them. So the first question that we have for you, Andre, is you had mentioned that one of your findings was that fungi was much more productive for protein expression than yeast was. And we're curious why you think that is. What are their specific production rates as compared to mammalian cells, which are on the order of 10 to 60 picograms a cell a day?
Yeah, actually, talking about fungi overall, that's their nature. In nature, they need to secrete a lot of enzymes to digest the food, the polysaccharides. Then is net they evolved a natural secretion system or very efficient secretion system. Then we are just trying to use this natural capacity to produce more recombinant proteins. And in this slide, I share it's possible to achieve 50 grams per liter or 60 grams per liter of recombinant or secreted proteins using fungi. And that's the major point, I would say. It's naturally, they evolved to secrete large amount of proteins. Yeah.
Interesting. Thank you. Another question here is around whether you have investigated how the introduction of new sites of N-glycosylation into recombinant proteins might affect secretion or production level and activity.
New site, actually, I show only one mutant. We try to create four non-native glycosylation site, and it didn't work. The protein was not secreted at all. Then I'm limited to this data, but if you look in the literature, you can redesign some proteins, adding new glycosylation sites and maintain the function or even improve the function. Then we can find in the literature many examples of this kind of strategies, inserting new glycosides. Yeah.
Awesome, thank you. There was a sub part of this question as well around whether you know if specific mannose structures might further affect artificial N-glycans.
Further mannose residues. We have seen that the position of the N-glycosylation sites is more important the composition of the glycan for function and secretion. I mean, then we can maybe discuss about immune system recognition and so on. But in the field of structure and function, changing the sites uh, can interfere more in the protein secretion than changing the glycan by itself. Yeah.
Got it. Thank you.
You're
Um,
welcome.
Another question here is around um, how there was a company named Dyadic International, founded in the 2004 timeframe, that had a similar glycoengineering technology with yeast. They appear to have a number of proteins in their pipeline, but their commercial success has been kind of lackluster. Curious if you know of this company, but basically they're curious. Is there a difference in their technology versus what you're developing, and will it have any kind of unique barriers to commercialization that would be good for us to be generally aware of?
Yeah. Actually, in the literature, we can find yeasts modifying to produce mammalian-like, real complex mammalian-like structure. It's more advanced in yeast than filamentous fungi, basically, basically because it's more difficult to manipulate filamentous fungi than yeasts. The genetics and the time to obtain mutants and to confirm mutants. In Pica pastoris, you can find the complete mammalian-like glycosylation inserted in the cell, and it's possible to obtain a high percentage of mammalian-like glycans. Then you need to add heterologous genes and complement the pathway to produce the mammalian-like glycans. But in filamentous fungi, as I mentioned, probably Professor Uffe's paper is, I would say, the most advanced. They obtain 80% of truncated mannose. Is very nice. But now we need to continue adding new genes, trying to achieve this. And talking about yeast, is easier to scale up yeast because filamentous fungi have extended morphology, high viscosity in the bioreactors, and so on. Then few companies dominate this, the fermentation of filamentous fungi. That I think this is a limitation. You know, have few data of scale up of filamentous fungi in the literature. You can find in some patents, but it's difficult to find real data, big fermentations in the scientific literature. Then, yeah, we have still have some bottlenecks, but at the same time that we can invest our science, yeah, to solve these bottlenecks.
Absolutely. So lots of opportunities for continued exploration on the scale up front. Thank you for
Yeah.
that response. Um, there is a follow-up question around whether you can speak to some of the most promising fungal strains that are available for commercial use. Also, okay, if you want to pass on any of these questions.
Yeah, probably the strains I mentioned, Aspergillus niger, we have used for more than 100 years to produce citric acid, for example, many enzymes. I'm intolerant to lactose, then every week I buy lactase from Aspergillus niger, for example. And Aspergillus oryzae is also used for food applications for many decades, and is also a very nice chassis strain for recombinant protein production. By the way, some companies are scaling up the production of beta-lactoglobulin using Aspergillus oryzae, reaching 30 to 40 grams per liter. It's huge. Then I would say these are the most important chassis talking about filamentous fungi, and they have the GRAS status. They are recognized as safe, long history of using food and so on. But domestication, I would say domestication of new fungal strains can also be applied for the future, you know, for long-term research. We have many new strains we can explore for these applications.
Definitely. Such a natural biodiversity, right? So rich.
Yeah, yeah,
Yet we've just scratched the surface of the strains that might be useful for
yeah,
this mission of building better meat mimics.
yeah.
Um, a few more questions here. So Joseph is asking whether, so the deletion of the glycosylation pathway genes showed different morphology for fungi. Did it change its growth rate as well?
Yeah, we didn't measure in a bioreactor system and a control only in airline layers in the lab. But yeah, the double and the triple deletion affected the fungal biomass. We obtain a lower concentration of fungal biomass after these mutations, which means that probably this mutant strain can have problems at the scale up, I would say. But we have no data to really show this, only in the small scale. And
Thank you.
that's the trade-off. You know, we can obtain higher secretion, but you need to take care of your cell because the cell is the biocatalyst, the most important part in the bioprocess. Then we
Hmm.
can avoid to affect a lot the cell physiology.
Hmm. So you had spoken to some of the complexities of working with fungi, the morphological complexities over working with yeast. Um, assuming one of the participants is curious. Um, what would you say are the additional advantages of working with filamentous fungi over yeast, assuming that you're able to actually achieve the same developments in both? Like you had spoken about the increased production rates, that was a benefit. But if you're able to achieve the same development in both filamentous fungi and yeast, do you see additional benefits to working with fungi?
Yeah, I would say the benefit it's the higher titer of protein secretion. The literature is clear. It's possible to achieve higher titers using filamentous fungi than this. That's for me, it's the main idea. But on the other hand, we have many other negative aspects, as I mentioned, but we can solve by mutations, including the morphology. We can improve this morphology for bioreactors, deleting some genes related to cell wall synthesis in fungal, in filamentous fungi, and so on. In the literature, we can find combination of mutations of the so-called morpho genes to increase or to improve this characteristic of filamentous fungi.
Thanks, Dr. Damasio. Yeah, it definitely feels like there's a whole lot of promise here, but we're in fairly early stages, and lots left to explore. Here's a question from Christian asking, how do you think the fungi would react to suddenly producing complex type N-glyc? It might recognize its own proteins as foreign, and in that case, it might also add negative charges like sialic acids that might interfere with function. So curious if you have any thoughts on this.
Yeah, again, we have many data from the literature, not using alternative proteins yet, but other recombinant proteins. And yeah, it can affect, first of all, fungi cannot sialylate proteins. If you want it, you need to add genes and enzymes, non-natural enzymes, I mean. That's the first point. And yes, because the charge of these glycans, the protein structure, and then function can be affected. Then I would say it's protein specific. That's the problem. If for humanization of recombinant proteins, probably we need to test protein per protein. Each one you need to study structure, function, secretion, and try to find the balance. Yeah, but definitely can affect protein structure and function.
Thank you. Two last questions here. One of the participants is asking if you can comment some more on the secretion of host cell related impurities that might be found in filamentous fungi.
Sorry, can you repeat, please?
Yeah, I'm not sure I fully understand this question either. Could you please comment on the secretion of host cell related impurities in filamentous fungi?
Probably impurities, you are mean other proteins in the secretome, right? And yeah, as I mentioned, fungi can secrete many proteins, not only your recombinant proteins. And again, we can delete unwanted proteins. That's a topic people are working for filamentous fungi. Delete proteins that compete with your recombinant protein for the secretion system. Yeah, then we can delete and that the secretion system can be more dedicated to our recombinant protein. And you can reduce these unwanted proteins by changing your culture media or adding some catabolic repression, such glucose, for example, is a catabolic repressor. Then we reduce the expression of many proteins that can compete with your recombinant proteins. Then it's a real problem for the downstream process, but we have tools to reduce this problem. Yeah.
Thank you, Andre. And last question here, and then we'll move towards wrapping. Um, a participant is wondering whether you can comment on the effects of glycosylation on protein allergenicity, which I think you touched on lightly, but um, if you can speak to that a bit more.
Wow, immunology is not my field.
Yeah.
I know a little bit about that, but I can mention that high-mannose can be recognized by human immune system. There are many works in the literature showing that. Once our immune system recognizes this high-mannose structure, this cell is pre-activated, and when you get in contact again, this system can react and generate allergenicity and so on. But as I mentioned, it's more common in the field of therapeutic proteins than food proteins, yeah, because therapeutic proteins are used in a purified form and it's direct contact with the immune system, yeah. Then there are few clinical trials showing this reaction for food proteins, but you can find clinical trials showing some allergenicity using proteins produced by microbial cell factors. Then we need to take care, and but at the same time, we need more data. We need clinical trials to confirm this. Yeah? But the history of using fungi like mushrooms and other fungi, like corn, yeah, using people consuming mycoproteins for decades, I would say it's safe, at least considering these examples.
Definitely. There's a demonstrated history of safe
Yeah.
consumption with
Mm.
additional health benefits. So, anyways, thank you for all
You're
of
welcome.
fantastic responses and for just engaging in this discourse, sharing your wealth of knowledge with this community. This has been a joy, Dr. Damasio. Is there anything you'd like to say?
Thank you so much.
Anything you'd like to say to wrap us up today, to our scientific community of alt protein folks?
Yeah, I can say that we need more people working on
Yeah.
filamentous fungal engineering, I would say, because there is a huge potential, especially at this moment. The market of recombinant proteins are increasing a lot, and these cell factors are very, very nice. But we still have few groups working on that. We need more literature using different strains, domesticating strains, and yeah, that's the I would want to say actually.
I love that. Such a brilliant call to action. I think they're definitely underutilized, and hopefully others here in the community, if they feel so inspired, can reach out and maybe this can spark some collaborations down the road. Thank you so much again, Andre, for sharing your wealth of knowledge with our community. Hope you have a fantastic rest of your day, and I hope the rest of you join us next month for our next Science of Alt Protein seminar. Thanks, everyone.
Thank you.
Thank you. Take care.